आखिरी बात वीडियो क्लोज है ऑडियो जी अस्सलाम वालेकुम स्टूडेंट वेलकम टू बायोलॉजी ऑनलाइन क्लास यू आर हियर विद सरफराज अहमद काजी हेलो अस्सलाम वालेकुम स्टूडेंट वेलकम टू बायोलॉजी ऑनलाइन क्लास विद काजी सरफराज अहमद
वालेकुम अस्सलाम सर
okay thank you for coming thank you very much students welcome to biology online class I just want to see how many people are here with me right now okay we have here with us Adil Zunera Ishal Fahad Hamna Hassam and Stephen so these people welcome to biology online class a brief introduction to you I'm teaching biology for the last 24 years here in Islamabad in different institute and Alhamdulillah, my students they are producing excellent results uh, in this area as well as abroad as well. So all these students, those who are producing good result, definitely because of their struggle and because of their effort. In addition to that, I would like to clarify this thing. Some of my students, they also in 2003, one student from City School, she got gold medal, Mahavish, and another student 2007, it was a regional distinction by the student at that time. And there are many more students every year. They gave 100% result in all the class in biology from 2000 to 2010 and onward up till now. And I also taught abroad in Manarat Al Riyadh, Saudi Arabia. And during the last 2 years, my student of all AS and A2 levels, they scored 80 to 100% results in the Cambridge as well as in the AP class biology. So all of these students, those who are scoring grades, definitely they are working hard. No, first of all, I would like to welcome all of you to biology IGCSE and GCE session in which, as you know, that your syllabus has some main areas which we will go through as far as the topics are concerned, syllabus is concerned, and the paper pattern and how you are going to attend the paper. Now these areas we will discuss the planning of the paper, how you are going to plan or in the specific period of time, MCQ paper you are going to how you are going to handle that, how to attempt the paper of theory, structured question paper, both GCE and IGCSE. And in case of paper 6, which is basically related to ATP, alternate to practical, how to draw the diagrams and these things we will discuss in this. Then there are some reviews at the end of some past paper latest question. And in addition to that, I would like to also specify that your physical and mental fitness is very important during this session, as well as during the whole month which is going on, because in the next month, inshallah, you are going to give Cambridge examination. So you have to make yourself fit and your routine in such a way of all your sleeping or feeding or all these things which you are doing and studying and study plan, and using all the resources relevant to the topic, and solving the past paper questions as well as the assignments which I will give it to you on the group as per requirement. Planning, I would like to tell you here in brief, because this time has with us up to 3 to 3:10. So planning is very important thing in your paper. First of all, I would like to suggest read all the paper first of paper 2 and paper, I mean to say theory paper or practical paper, ATP paper. Your theory paper, when you're getting it in the exam, whether you are in GCE or IGCSE, read all the question first, then start doing the question. Before not doing start from question 1 part A and start it. No, read all the question first, then start doing the question. It will organize you with respect to your topic which is in the question coming. And how you for as far as the questions are concerned, in the middle of the or in the beginning of the or at the end of the question, how you are going to handle, please, you should have complete chapter in your mind when you are dealing any question, because mostly the questions are relevant to one or sometimes two topics together, or it's a merger of one or two topics. Another thing which is important related to paper 2 as well as particularly paper 6, that is 62, which we call 62 in GCE or IGCSE both, and that is practical paper design. This, in case of practical paper, you have 2 or 3 questions which are related to, one of them is related to your graph, your diagram, your labeling, and second is related to your magnification plus your analysis question, your analytical question related to analysis question, in which you are going to do some parameters are given to you, which we'll discuss at the in the coming second session of this workshop today, or in the third session of this workshop today. Now, I would like you to, when you are revising, you have to please, whatever you are reading, first revise it by writing practice or by oral practice, and write the main points. Then your overall timing should be for each and every topic or each and every subtopic on which you are working. It should be according to that, or because you have to finish all the work in your planned time. Not only, definitely you are going to go for biologies, you have other subjects as well. So specify your timing for that. In case of paper, when we go for, remember, if it is of just I'm taking you afterwards your delivery of the answers in the past paper question. We will do some of the practice here and the coming paper, but right now, how to solve the question. Be active and very much, when we say in your answering the question, you have to be very much focused to the command statements. If he is asking for description, try to go for description according to the graph or table or according to the information given into the paper. Secondly, if he is asking for explanation, please try to give the concept in that. Full-fledged knowledge related to the concept in that area, try to give it. Try to go through your finishing your question. After that, try to go through it quickly, and all your book knowledge or your reference book knowledge should be expressed in your answer, which will take you to a star or inshallah towards distinction. Now, as far as this is concerned, no assessment as far as our paper 1, 2, or paper 1 MCQs in the same way. Next paper is structured question paper. If you are from GCE, it is definitely 5090 paper 2, or if you are from IGCSE, then it is 0610 paper 42. So in that cases, particularly try to go with each and every question step by step, compile it. If you feel some of the area is requiring more time, so please try to cover it, otherwise move to the next question, because do it at the end otherwise. This is the only option which we have. Now we are moving towards the topic session right now. It is a brief introduction about our workshop beginning. Now, as we enter into GCE and IGCSE syllabus, and you are familiar with the syllabus very well, in the last 2-3 years you have gone through. Now I'm going to compile the main areas which are important in chapter 1 related to characteristics and classification of the living organism. As I told you, there are three parameters. There are three papers you have. In case of IGCSE, there is paper 2, that is 45 minutes. Paper 4, that is 1 hour 15 minutes, 80 marks, and paper 6, that is of 40 marks, paper practical. Now, starting from first chapter, brief introduction as far as the characteristics are concerned. Sometimes 1 marks, 2 marks question in the form of table or chart is given. Movement, respiration, sensitivity, growth, excretion, nutrition. In that case, just give the specific definition for that. Movement, when we say it is the change of, when we say change of place. Respiration, you will specify aerobic and anaerobic, with oxygen and without oxygen in case of respiration. Mention this thing clearly. Sensitivity, response towards a particular stimulus, for example, temperature, so on. In the same way, excretion, removal of nitrogenous metabolic waste material, specify in that case pertaining to your excretory organs related to kidney, lungs, or skin. Next, nutrition. Nutrition, our food nutrition, our requirement of food by the body, macro and micronutrients. Macronutrients are carbohydrate, fat, proteins, which gives energy. Remember, and micronutrients, they are not providing any energy. Vitamins, minerals, and fibers. They are the part of enzyme as a coenzyme. Remember this thing. Now, as we move on in this main area chapter, that is, sometimes the question come about for arthropod classes and the basis of their distinctive features. Most important feature which they are asking in the paper, that is crustacean. Arthropods are divided into four main classes, that is crustacean, arachnids, myriapods, and then insecta. In this case, you have to remember the most important thing, that is crustacean have more than 4 pair of legs. Arachnids have 4 pair of legs. Myriapods particularly, they have centipede, millipedes, remember. And insecta, we have 3 pair of legs. This arthropoda is included in case of IGCSE syllabus. And as we move on, okay, now in this regard, next, some short questions relevant to 1-2 marks relevant to virus, bacteria. Remember, bacteria is a cell. It has a cell wall and a plasmid DNA, circular plasmid is there. And in case of virus, it is not a cell. It has an outer protein coat, or it has, when we say, internal DNA or RNA. If you see in this case, this is related to this diagram which is shown you here. It is just the example of different viruses which we have currently. In case of COVID-19, it is just like that, or even HIV virus, it is just like that. Outer protein coat, internal DNA or RNA. So they are living because they have DNA, RNA, and they are non-living because they can be crystallized. So sometimes the question based on that coming to differentiate between virus, bacteria, or fungus, or fungus with, we will do in the coming topic about fungus and planta and animalia kingdom. Now, right now, related to cell structure, question based on there are different cells which have similar function but different structure. Like red blood cell, it transport oxygen and carbon dioxide. As you can see, with oxygen it forms oxyhemoglobin. With carbon dioxide, which is transport inside the blood in the form of H2CO3, that is in the form of carbonic acid, or also in the form of we can say inside the blood it is transferring in the form of carbonic acid. So it is transporter of oxygen and carbon dioxide. No nucleus, hemoglobin is present, and it takes approximately 3 to 500,000 hemoglobin inside a single red blood cell. Then muscles, when we go for, they also contract, relax, so they also move the person. So there, when we say, as far as the transporters are concerned, red blood cell transport oxygen, carbon dioxide. Muscles transport the person, or we can say move the person in this way by contraction and relaxation. Ciliated cell, this is inside the trachea, bronchus, bronchiole. They have small cilia, hair-like projection, which is removing the mucus out, or so they move upward and downward with the help of energy. Root hair cell also a specialized structure, large surface area volume ratio inside the plant. It transports glucose, amino acid from soil into the into the root hair cell. It has high amount of mitochondria, which provide ATP for active transport, and also diffusion takes place. Xylem cell, it is a dead cell. It transport water and mineral. Remember, student make a mistake mostly here. Here, diffusion, active transport, things are not going on. Particularly the thing which are going on here is oxygen. Is water is going with the help of osmosis. Remember, and minerals are going with the help of simple diffusion. Remember, it's a dead cell. So water moves upward in the form of, we can say, capillary action, which we'll go through in the coming topic, plant section, transport in plants or nutrition in plants. So capillary action is the force of water molecule between one another, or you can say it is the force of water with water. So this is known as cohesive force, which is going to take water upward. And if this force of water interaction with the walls, we call it as adhesive force. Water interaction with the walls. So this is adhesive force. So in this way, water moves upward inside the xylem vessel. Remember, lastly, we have palisade cell in this chapter, which is in progress going on. This is inside the leaf section. Remember, there are three main cells in the leaf section, which is photosynthetic in nature: palisade mesophyll cell, spongy mesophyll cell, and guard cell. Palisade mesophyll cell, spongy mesophyll cell, all of them, they do photosynthesis. Remember, because they have chloroplast. Chloroplast is green because of chlorophyll. It contains magnesium in it. Due to that, it is green in color. So this is your main area related to different cells with similar function. Overall, they are transporter. Diffusion, it is the movement of substance from high to low concentration. In case of digestive system, in case of root cells, substance move from high to low concentration. And it does not require energy. Remember, there are different factors which should be also in diffusion mentioned in your answer. It should be mentioned. Large concentration, fast diffusion, temperature increase, also increase diffusion, more surface area, also increase diffusion. Next is osmosis. It is a diffusion of a water molecule from high to low concentration through semi-permeable membrane. Remember, in case of animal cell or in case of plant cell. So questions are based on that. Sometimes they are coming related to osmosis, and they are taking you just to highlight here one of the parameter. I'm sharing one of my slide of lesson with you right now. Now take a look of this short 1-minute animation.
Many perfumes and medicines move by diffusion. Here a crystal of purple potassium permanganate spreads into water by diffusion. The particles are moving and mixing with the water molecules all randomly by chance. Osmosis explains a great many things in biology. We're going to show osmosis in action. Here is a semi-permeable membrane. It allows small molecules like iodine through, but not large ones like starch. Remember, when starch and iodine mix, a blue-black solution is made.
Okay, look into this student, look into this practical which is going on in front of you about diffusion. Starch molecules are bigger molecules. They cannot move out of the tube, but outside the molecules, they can diffuse inside, for example. So if you are getting a question relevant to that, take a look. I am taking you to the a question right now, which is recently given in the paper. Look at this question. They specify in 2020-21, they gave a question here right now. You are adding a sucrose solution in the whisking tube and putting a knot at the bottom, remember? Now, after that, you are placing it inside the distilled water. Definitely, when you're putting inside the distilled water, so sucrose solution, they will not go out because it's a disaccharide, large molecule. It will not move out from the whisking tube, which is semi-permeable in nature, as you know. So water molecule from outside, they are small in size, so they can move from outside to inside, and due to which water table rises inside. Now, this is the main concept. Now, what is asking in this question as we proceed further? As for sucrose, DW for water, distilled water, because he has taken two sets of tubes. Now, take a look. After 0 to 15 minutes, he is showing you here right now, there is an increase of level inside the whisking tube. And here, the control experiment, before and after, because there was distilled water, after there was distilled water, there is no change on the right side. As you can see in the right side diagrams, the level is same. There is no change in the level before and after. But in case of in case of sugar, the sucrose solution, the level has gone above. So because water entered inside, so further it increased the level. Now we are moving to the next part, what he's asking and how he's asking the question in the paper. Here he's asking to make a table. So we will make a quick table here in case of just to make a table with respect to here, and putting here some parameter, a sucrose solution, distilled water, and initial length, final length, and you are making the parameters here in both the cases. And what is the difference in that? You will specify, and at the bottom you will show the difference in this. Here you will put the difference of the sucrose solution and distilled water. You will put the figure by using the ruler in that case. Now, as we move on, how he's asking question further. Okay. Length, you will simply measure the length of the distilled water, measure the length of the sucrose solution. Here you will put it. Remember, all the length which you are measuring in ATP paper or in theory paper, try to, even in MCQs, you have to measure in millimeters, not go for centimeter or meters. Now, why is it important to take the measurements from the same place in the dialysis tube? For definitely for accurate reading. What are the conclusions we can draw from this experiment? Definitely small molecules, they can enter into the whisking tube, but large molecule cannot move out like sucrose into the water. From whisking tube into the water, it cannot diffuse out. So identify the variables we have in case of, remember, independent variable is manipulated variable which can be changed, or it is the cause of the experiment. So you can change different concentrations you want, but here right now he's asking one set of result collecting data during the investigation. Why it is better to take several sets of data? You can take 3, 4, 5, 6 reading, definitely for more accuracy, for more reliability. So you can make a mean of that and collecting, or we can say a very precise answer of your practical or your research, whatever you are doing in any practical. Okay, suggest, okay. As for now, this is about this question which was relevant to that topic. Now we are going back with the same. We have finished this animation right now and moving to the next part that was relevant to diffusion osmosis, a topic which was under progress going on. I hope so. Everybody is watching. Hello. Everybody is watching the slide. Please confirm.
Yes, sir.
Fantastic. Okay. Now we are here back with the diffusion osmosis topic, which is in progress. No, osmosis for water, we have done one of the practical element to that. No, active transport. Remember, active transport is the movement of molecules from low to high concentration or against the concentration gradient. It requires energy or ATP. Uh, in this case, you will specify the example of your root hair cells, of your own body villus cells, the glucose molecule, amino acid molecule, they enter into your body or in the root hair cells by active transport. If they go from low to high concentration, they require energy. Mostly the questions are coming on that. They are also asked in the paper. Excuse me. Uh, Biological molecule. In that we have carbohydrate, fat, proteins, which we will go. Then vitamins, two main vitamins, and minerals, two main we will go through, and importance of water. Is asking for the starch test. Iodine with starch, we get blue black color, as you know, we have done just recently. Reducing sugar test, very important test. Practicals in the paper, they are asking the practicals. What is the difference between reducing sugar and non-reducing sugar test? I'm just taking you to right now with one more short animation, so you can be able to see that how we are doing that reducing sugar test in case of reducing sugar test. Basically, when we make glucose with Benedict solution, the test is done, and in this glucose and Benedict solution, they are going to uh, place inside the test tube, and that test tube is placed inside the beaker, and then we are going to heat it up to 80 to 100 degrees C. So if the color change from blue to orange red, if the color change from blue to orange red, it's mean glucose is present, and if it is not, then it's mean glucose is absent. Now there is a short animation related to one of the food tests shown here: protein, fats, and carbohydrate. We will go quickly just to make a quick review of that all in short. Yes, everybody is watching the slide. Please confirm.
Different types of food
Yes, sir.
Thank you.
make up a balanced diet, so it is useful to test for the different food types. Proteins are important for growth and repair. To test for protein, we use BIOLOGYTRIPS solution. Protein turns BIOLOGYTRIPS purple. Sugar gives us energy. To test for sugar, we add Benedict solution and warm gently in a water bath. Sugars turn Benedict solution.
Remember, it's very important when you put the glucose inside the test tube and add Benedict solution, place this in the water bath. It is better. If you handle it directly in the hand, it is very dangerous. So try to be very much careful as far as the safety precautions are concerned. Your gloves, your goggles, your lab coats, because Benedict solution or even BioLogYTRIPS solution, they are highly, and all the solutions or the chemicals which are, they are highly corrosive or inflammable. So when you are putting this in the test tube inside the beaker, try to be make the face of that towards the other side. One thing more which I would like to highlight here as well. Look at this. I'm taking you to the whiteboard right now. For instance, you have a beaker right now, and you are simply a tripod stand is here, for instance, and the Bunsen burner is at the bottom, and you are placing a test tube inside this. This test tube, which is containing inside, for example, this test tube is containing glucose and Benedict solution. So when you heat that glucose and barium solution, remember you will heat up 80 to 100 degrees C. Remember, from 80 to 100 degrees C, it is important. If you will simply write warm it, it is not sufficient. So you have to heat it from 80 to 100. I'm writing in short degree C, and you will see the color change. It will be from blue. First of all, I'm writing in short blue to blue, green, then so it will change the color from blue, green, yellow, orange, and red. So why this color change in this sequence, just like a rainbow? The reason is this: if it is blue, it has less amount of glucose. When it is more reduced, it will convert into a little green and then greenish yellow, and ultimately this color further increase to orange. And if all the glucose is reduced, it will become brick red. So we can make some figures as well. For example, it is 0.2 and this is 0.4, for instance, and in the same way this is 0.6, and in the same way this is 0.8, and last one is 1.0. So high concentration of glucose in red color. So this is your test of reducing sugar. If it is sucrose, remember, it will remain blue, it will not change. And in that case, there is a tricky question come in the paper. This sucrose, because sucrose is the combination of glucose plus fructose. It's a disaccharide. So what you will do, you will add in it HCl, or remember, or you can add in it enzyme. They will give you this option as well. You can add HCl, or you can add enzyme which break the bond of glucose fructose. Ultimately, glucose is separated. When this glucose is separated, remember, when this glucose is separated and you again heat it with Benedict solution, it will give you a brick red color. Remember, this is for non-reducing sugar. Remember, that is sucrose. You are going to, if you heat, it will blue to blue, no change. But if you will add in it HCl or enzyme, they will break the bond of glucose fructose, and in this way they will be separated. Glucose fructose will be separated, and the color again change if you had with Benedict solution. So this is about the briefing about the reducing sugar and non-reducing sugar test. Ooh, just a minute. Just to confirm, everybody is watching the slide right now of the test, food test.
Red.
Please confirm everybody is watching the slide of food test. Students? Hello?
The screen recording is not working.
Okay. Screen recording is not working. There is something wrong going on. Okay. Just a minute. Screen sharing नहीं हो रही. मेरी screen share नहीं हो रही. For example, अब मुझे share करनी है ना, वो किसे share करना है? वो कहाँ नाइट्रो कहाँ? Where is that? Post ने पीछे से कुछ किया. इसको करें. Oh yeah, yes. अब screen sharing आ गया. पीछे से ऑन करनी पड़ेगी. Yeah, right. Thank you so much. So we were here with the reducing and non-reducing sugar test. Reducing we have done, that was about related to glucose with Benedict solution. Students, are you watching the slide again now?
Sir, we can't see anything.
Just a minute. Okay. Up करेगा. That's fine. Thank you. Right? I want to show this one. Everybody is watching the slide right now. We are back here with the food test.
Yes, sir.
Oh, thank you so much. Now, this is for reducing sugar test, protein test, and fat test, particularly. In case of fat test, emulsion test, it is known as, in which 3 ml we are adding inside the test tube oil and 3 ml alcohol. It will become transparent or it will give white emulsion, which confirm, remember, the presence of alcohol in the food test. Now, I'm sharing once again continuity of the same experiment. Take a look. Are you watching the slide?
Starch also gives us energy.
Yes, sir.
Okay,
One
thank
drop
you.
of iodine turns blue-black in the presence of starch.
Now you can see there are 3 samples shown. And first, for example, mango. In the second, sugar. Third one is noodles. And any one of them will become blue-black. It means starch is present. This is for the starch test. We have just compiled that. Now we are moving further for the fat test.
Fats are concentrated energy foods. Here, we have cooking oil in a boiling tube, to which we add pure alcohol. The fats dissolve completely in the alcohol.
Remember this thing, when you are adding fat with alcohol, it will become transparent, or sometimes it will give white emulsion. Now, there is another test which is also sometimes come in the paper about vitamin C. Vitamin C in orange, lemon, as you know. And if we are going to add that in the test tube and drop by drop, we are running the DCPIP. It is a colored solution. If we add vitamin C inside the DCPIP, it will become transparent, which confirm vitamin C presence. If it is completely transparent, it means high content of vitamin C present. If it is not, it means less concentration of vitamin C present. Now, in this chapter, they also mentioned something about DNA. In brief, DNA is deoxyribonucleic acid. Detail in this in case of inheritance chapter. But right now, you should know at least the basic sequences of DNA, and that is, remember, that is always वो लिख नहीं रहा है. It is not writing. देखिए, पीछे से होने के होस्ट को मुझे ऑन करते हैं वहां से. देखिए ना, ये I want to write something. I want to type something here. It's not typing anything. Okay, just to देखिए कुछ लिख नहीं रहा, मैं मार्कर से भी लिख रहा हूँ. It is not writing anything on it. Okay, just to brief students, welcome back. DNA always combine. Remember, DNA has 4 main bases. 4 main nucleotides which are combining in a sequence. A always combine with T, adenine with thymine, remember, and guanine with cytosine. So adenine, guanine, cytosine, thymine, they are 4 main bases, but adenine always combine with thymine, remember, and guanine always combine with cytosine. Now we are proceeding further about, as you can see here, ATGC combination in DNA. Enzyme. Enzyme, the question sometimes it is coming in the paper, describe, define what are the factors affecting enzyme. pH, temperature, concentration, all also they specify what is the mechanism of enzyme action, that is lock and key. Remember, enzyme is the lock, substrate is the key, and they make a lock and key complex. Ultimately, they form a product at the end. Enzymes overall, it can be used again and again. Remember, enzyme, sometimes they ask about the property of enzyme. Enzymes are made up of proteins, they are reused, and they are affected by temperature. Okay, enzyme. You are watching the slide after DNA. As I told you about ATGC combinations. Students, confirm
Yes,
please.
sir.
Thank you. So after ATGC combination of DNA, we are here with enzyme. Enzymes are, okay, thank you. Enzymes are biological catalysts. They are made up of proteins. They are reused. They are affected by temperature, pH, and concentration, remember. And in the same way, you will specify their mechanism of enzyme action. When we say mechanism of enzyme action, in that case, remember enzyme work by the principle of lock and key mechanism. So lock and key. Enzyme is the lock, substrate is the key, and ultimately they make enzyme substrate complex and the product is formed. Now, these questions sometimes they are asking about effect of temperature. Remember, at every 10-degree increase, enzyme action become double. Enzyme action become double. So, for instance, मैं लिखना चाह रहा हूँ उस पे लिखा नहीं जा रहा हाँ ठीक हो गया that's right thank you it's okay thank you now as we move on as we said that at every 10-degree increase enzyme action become double. For instance, here is we have effect of temperature shown, for example 0 in the same way 10-degree or 20-degree, just a minute, 0, 10, 20 or 30 or 40, for example, in the same way 50 or 60 and so on. So remember, at every 10-degree increase, enzyme action become double. They are asking something about optimum temperature. Students do a very, very big mistake here. Try to be very careful. Optimum temperature, when we say it is, basically it is 35 to 40 is the optimum temperature. Remember, it is the optimum temperature. Here you will specify, this is the optimum temperature range. It's mean what? It's mean that all the active sites of the enzymes are full. It's mean maximum substrate is converting into product. It's mean active sites full, substrate converting into product. It's mean the kinetic energy of the enzyme substrate interaction is very high, or collision forces are very high. This is the meaning of the best temperature. People write optimum mean the best. It is not sufficient, please, for A grade or A star or distinction. Try to be very much careful in that. Same is the case for pH range. Optimum temperature mean when all the active sites are full, substrate is converting into product, and kinetic energy is high. In this way, you will handle this. Sometimes they are asking question relevant to, in the past paper, they are asking question in this way that some washing powder, some, you have seen washing powder with different color, blue granules, green granules, red granules. They contain inside lipase, amylase, protease. And they ask a question inside, for example, you are placing, there is a blood stain on some clothes, or there is some fat stain on some clothes. So you will specify your answer very clearly, because enzymes are specific. So lipase act on fat. In the same way, protease act on proteins, or amylase act on starch. So if it's a blood stain, you will specify protease is present inside the granules of washing powder that is going to act on the blood stain. And as far as the quantity is concerned, they will ask sometime that why the stain is not removed, because the quantity is less. So, or maybe the temperature is not appropriate. Maybe the pH is not according to the acidic condition, because proteins work better in the acidic condition. These parameters are mostly asked related to enzyme, remember. Now, as far as nutrition is concerned, plant nutrition is concerned, remember, whenever you are getting question about photosynthesis or respiration, never ever forget to write the equation. Carbon dioxide plus water, glucose and oxygen. Carbon dioxide from the atmosphere, it enters into the through the stomata of the, we can say, guard cell, into the leaf, and water is entering through the xylem vessel from the root stem up to the leaf. And due to other factors in photosynthesis like light, chlorophyll, as well as, we can say, temperature, which is not mentioned here, it is also very important factor. And this is going to build up these 5 factors affecting photosynthesis, building up glucose in the palisade cell, in the spongy cell, in the guard cell as well. So oxygen is a byproduct which is produced, remember. Now, as we move on, other than after this equation, mostly the questions are asked, describe the explanation of investigation why chlorophyll is necessary for photosynthesis, or why carbon dioxide is necessary for photosynthesis, why light is necessary for photosynthesis. Remember, for that purpose, you have to Destarch your potted plant on which you are working. You will take a potted plant or take two potted plants, one as a control maybe, or you can put that potted plant in the dark for 48 hours. After putting it into dark 48 hours, remove the plants. And then you are going to test for that. Before that, we enter into light necessary for photosynthesis, carbon dioxide necessary for photosynthesis. Any experiment, we have to destarch the plant. Remember, in all the cases, what is the limiting factor? When all the factors, look at the above equation, is available and they are optimum, and any one of the factor is in scarcity, for instance, for example, carbon dioxide, it's mean that is in limiting factor. All other factors are increasing continuously according to the experiment which is in progress, but any factor which is in scarcity, not increasing with the other factors, it means that is a limiting factor. Just if you take any example relevant to enzyme action, even temperature, pH, or concentration, all of them you have optimum, but any factor is in scarcity, it means that is a limiting factor in the previous enzyme chapter case, which we have recently gone through. Now, as we move on, remember in your investigation of chlorophyll, carbon dioxide, and light is necessary for photosynthesis, please specify at the end conclusion after the experiment, after testing it for starch with iodine, okay, it is present or not. Same is the case carbon dioxide, just to make a quick review as well. Here you have two potted plants covered with the bell jar, one containing sodium carbonate, which provide carbon dioxide to the plant, other contain potassium hydroxide, which is absorbing carbon dioxide. So that is a control experiment, which is going to absorb all the carbon dioxide. But Na2CO3 or NaHCO3, it is providing carbon dioxide. It is fulfilling all the factors. So plant A will, if it is tested, the leaf will be tested, it will become blue-black because all the factors are fulfilled. But in case of plant B or potted plant B, which is covered with the bell jar, and it will become starch is not present because carbon dioxide is absorbed by potassium hydroxide. So it will remain yellow brown. So conclusion should be very much clear in your answer. Leaf structure labeling, sometimes it is asked. Now take a look of this question, leaf structure. Mostly, just to take you to one of the, just a minute. Depending upon whatever the question they will give you about the limiting factor, water, carbon dioxide, temperature. So remember, if it is in scarcity, if it is not available, that is a limiting factor. This is the meaning of limiting factor. We have given some example here, light intensity. We have given some example of carbon dioxide here in this as well as temperature. Now, as we move on about the leaf structure, remember first layer, they will ask you to label the structure in case of paper 6, cuticle. Cuticle, waxy layer, which fix the light which work as a prism, and we can say light can pass through. Then upper epidermis has no chloroplast. Remember, it's a protective layer. Palisade mesophyll cells, they have 70% of the chloroplast. Remember, it's very, very important that palisade cell, they have 70% chloroplast. So they are going to do maximum photosynthesis. Secondly, spongy mesophyll cells. Spongy mesophyll cells, they have around about, we can say 20% chloroplast, and they do less than photosynthesis, less than palisade cell, and even the guard cell. Remember, the guard cell also have chloroplast. It is approximately 10% of the chloroplast. So all these cells, maximum, they are going to capture light. Palisade cell tightly packed, no air spaces. Spongy mesophyll cell, they have air spaces. Remember, here questions are asked, especially for A-grade student. Transpiration starts here. The palisade cell, spongy cell, they release water into the air spaces, spongy cells. Now transpiration from the leaf surface, first from the mesophyll cells, the spongy mesophyll cell, then from the leaf into the atmosphere. Remember, this is very, very important area, which is nowadays they are asking. Xylem cell, phloem cell, another part of this leaf, as you know, vascular bundle. Xylem is a dead cell inside the vascular bundle, transport water and mineral. Phloem is a living cell because it has two parts, companion and sieve tube cells, which transport sucrose, glucose, amino acid, fats as well. So their transport assimilates. So as far as the opening and closing of guard cell is concerned, very important question again. Guard cell, when they become turgid, remember, when the water is entering into the guard cell, it becomes turgid. From all sides, water enters into the guard cell, and these guard cell will become turgid. Due to their turgidity, they have a bean-shaped, kidney-shaped cells, and automatically inner lining is thicker and outer is thinner. So develop the pore build up inside, which is bigger in size, and that pore is known as stoma or stomata. But when water is not available, then what happens? Guard cell loses water, exoosmosis takes place, and due to the decrease of water potential, the guard cell become flaccid and pore become closed. Mostly it is happening in the night. Very less amount of gas exchange also continue, but 90% we can say the stomata is closed. Remember. Now, after this, as far as xylem movement is concerned, xylem water viscous movement, as I said, capillary action, cohesive force, adhesive force, fluid transport, glucose and amino acid. Some question based on the nutrition in plant chapter, importance of nitrogen and importance of magnesium. Remember, nitrogen is excuse me, nitrogen is necessary for growth and development of the plant. And remember, they asked you in which form it is going to enter into the cell. They enter into the cell in the form of ions and molecules, nitrate, or in the form of nitrite, ammonia, ammonium. And they are necessary for growth development. For even every cell require DNA or RNA. So for their DNA, RNA formation, nitrogen is necessary. Same is the case magnesium. It is necessary for plant leaf photosynthesis, chlorophyll formation. So magnesium is necessary for chlorophyll formation, chlorophyll is necessary for chloroplast formation, and chloroplast is for photosynthesis formation. Now this is related to importance of nitrogen. Now as we are going to the next, human nutrition, in that case, sometimes the topics start with the teeth structure. And as I told you about carbohydrate, fat, proteins, monodipolysaccharide sources, and the proteins, amino acid. Remember, the smallest unit of carbohydrate is glucose, according to your syllabus. Proteins, amino acid. Fat, fatty acid, and glycerol. Vitamin C and D in your syllabus, C in the lemon and oranges, and their deficiency causes scurvy. Remember, and vitamin D is also related to this present inside in milk, as well as some fruits and vegetables. They also necessary for bones and teeth formation. So if their deficiency, it will result into rickets, particularly with respect to if we go for calcium and iron deficiency. Remember this thing. So calcium deficiency for bones and teeth, iron deficiency result in anemia in case of red blood cell, and less amount of red blood cell producing hemoglobin, less oxygen transport, definitely suffocation, breathlessness. Fibers are in case of roughages, fibers, vegetables, which are not providing any energy, but they are the part of the balanced diet. So when we say balanced diet, it contains all the nutrients in the proper proportion. So fiber is also one of them, which is helping in peristalsis, bulkness of the diet. Water is the best solvent because it can dissolve solutes inside that. As we have discussed about the deficiencies areas, human elementary canal. Look into this question shown here. 5 main definitions sometimes ask: ingestion, digestion, assimilation, absorption, egestion. So ingestion, taken of food. Taken of food is ingestion. Digestion, it is the physical and chemical breakdown of food. Egestion, it is the removal of food which is in the form of feces. Undigested food which is removed from the body, that is egested, or we can say egestion or defecation. Assimilation, the digested food become part of the body. They will ask you to label that areas in the paper. Remember, just to take you after this directly to the question. Mouth, here they will enter into the question related to teeth sometime. Different types of teeth. To look at the right side, incisor, canine, premolar, molar. Incisor, broad top, canine, pointed tops. Premolar and molar, they have 2 cusps and 4 cusps. Remember, if it is a premolar, it has 2 cusps on it. And if it is a molar, it has 4 cusps on it. Mean to say they have 4 ridges on it. Just to show you, if it is a premolar and it is molar, So what is the meaning of two cusps and four cusps? So these are the two cusps. It has two ridges. And when we say four cusps, so it will be like that. It has four ridges or four, you can see, portions, which is basically increasing, which is used for grinding and increasing the surface area and volume ratio of the food stuff. Now this is just a minute. In case of teeth structure, so sometimes they're asked to label that, crown the top line, the top area which is made up of enamel and dentine, root the base with the cement. Inside there is pulp cavity which contain blood vessels and nerves, sensory and motor nerves. Blood vessels mean arteries and capillaries and veins which supply food, mean to say glucose, oxygen for the teeth growth. So if there is any kind of, we can say, plaque formation, they will ask you sometime. So plaque can be here on this place or on this place. Unhygienic food, when we eat, it contains bacteria, and that bacteria release acid, and that acid erodes to the surface from the sides or from the middle. Remember, and if this reaches to the pulp cavity, they will cause a pain. And person have to go for root canal, and ultimately further filling up. Sometimes we have to go for fillings. Maybe you have also experienced about filling. Now, next is in the digestive system, important areas which are digestion in mouth. As I said, amylase is acting from the amylase salivary glands on the carbohydrate and breaking it into glucose and amino acid, glucose and glucose, and which is start digestion from the mouth. Remember, because glucose digestion start from the mouth. Then esophagus, negligible digestion going on, although the previous is in progress. In the stomach, pepsin in the presence of HCl, it become active. This pepsin act on protein, break it into polypeptides, and here the food is in the form of chyme. HCl help into killing of a microorganism. It also helps to make acidic condition for enzyme pepsin to become active, and it also we can say internal. The question sometimes come tricky question: why the acid is not digesting the wall of the stomach? Why the pepsin is not digesting the wall of the stomach? Because inner wall of the stomach is highly or thickly lined with mucus cells, goblet cells. They produce mucus. That's why HCl, this pepsin is not able to digest that. So those people who have less amount of mucus or less water available in the mucus, ultimately they can cause problems like stomach cancer, stomach ulcer. They have some issues, but normally mucus is protecting the person from stomach from pepsin digestion, so that because mucus is produced. Then stomach, after that there is small intestine. Small intestine has millions of villi and microvilli, which are the organ of, remember, which are the organ of absorption. Questions are mostly based on that. Look at that. It is a single structure of villus. Inside there is lacteal. These lacteal directly absorb fatty acid and glycerol. Remember, but outer lining is absorbing glucose and amino acid by diffusion and active transport. They pass on into the veins and directly to the heart. Remember, pass on from the vein first. First they go to the from the vein, hepatic portal vein. I'm writing it short. Hepatic portal vein to the liver. To the liver. And from the liver, they go through hepatic vein to the heart. Remember. So this is the mechanism of by which we can say the pathway of glucose amino acid. They move from villus into the hepatic portal vein and ultimately liver. They will ask some question based on that. Why the concentration of glucose is high or amino acid is high here? Here, hepatic portal vein, glucose high, amino acid is high. But after liver, because liver is storing glucose in the form of glycogen function, liver also do deamination. So here there is high level of urea here. Remember. So I'm writing it short. Glucose is high, amino acid is high, but here high level of urea. So this high level of urea after liver in the hepatic way, and it has high level of, you can say, carbon dioxide. These questions are asked sometimes. What is the function of lacteal? Lacteal absorb directly fatty acid and glycerol into the lymph vessel. Fatty acid, glycerol, they enter into the lymphatic system, which is basically pertaining to immunological defense, or we can say which produces lymphocytes, also stores huge amount of fatty acid and glycerol. Remember this thing. Now, as we move on, according to the plan given to us, students, I have a very short uh, quiz made for you. I'm sending it to you uh, on your group right now. Please work on it, and after 10 minutes, tell me how much you have done. Right? Ji, everybody's watching the quiz question, please. Hello, students, are you watching the quiz question? Okay, thank you. No, there are short 20 MCQs which is taken, and you have that questions as well. Please try to go through it, and I'm giving you 5-10 minutes. Try to go through it and send me your answer in the form of WhatsApp snapshot. So I will see how much speed you are going to go with that. Try to finish one MCQ in 1 minute. Take 5-10 minutes. Okay, do maximum as much as you can. क्विज टेस्ट भेजना है ग्रुप के ऊपर, तो वो होस्ट ने मुझे रोका हुआ व्हाट्सएप पे। मेरा तो व्हाट्सएप खुला हुआ है, देखिए ये मेरा व्हाट्सएप है, लेकिन मैंने ये क्विज टेस्ट इस पे भेजना है। हाँ जी, व्हाट्सएप के थ्रू भेज रहा हूँ ना। थैंक यू सो मच। ओपन करेंगे तो मैं भेजूंगा। Gee, student, are you working on this worksheet which has some MCQs in the beginning? Done. Thank you so much. Gee, student, please confirm. I send you one of the short MCQs sheet on the group. Please check it. Try to check it on the WhatsApp group, please.
Yeah, sure, it's there.
Okay, thank you. Look at that. It has 20 MCQs. Take some time and try to go through it. We will discuss after 5 minutes. As much as you can make up. Okay. Basically, this is no doubt. This paper is taken from IGCSE. I also, okay. I know there are some students with us also from GCE. So I'm also sharing with you some question relevant to GCE as well. Would you like to mention in the group, please, those who are taking GCE, so I can be further specify that questions as well. Those students who are taking GCE, I am just uploading 5090 question 2 with MS and question 6 with MS, the latest paper which is here right now. Just to take you people with us as well, take a look of this paper, which is from 5090 now. The student, we have session 2 which is compiled now, and please, if you have done your MCQs, I hope so you have done it, and you can send it to me on WhatsApp separately, or you can just give us a snapshot. Otherwise, we are discussing it right now because of the scarcity of time is here. And now I am opening again. The GC student, I also sent paper 2 and paper 6 latest. We will discuss that question as well. Now I'm just discussing the first worksheet, first quiz sheet, which I have sent it to you on the group, starting from the last to the first, so we can be able to group up quickly with this worksheet as well, because we are running short with the time sometimes if we are not going with the pace as well. Now look at this question, starting from 20 backward. Here, everybody's watching the quiz sheet 1, that is MCQ number 20. Everybody's
Yes,
watching. Fantastic. Thank you. The photomicrograph shown is a human blood cell. Remember, the blood component carried out the process of phagocytosis. There are 2 types of WBCs. One is phagocyte, other is lymphocyte. Phagocyte engulf bacteria or pathogen and destroy. And lymphocyte, they produce chemicals or antibodies and they destroy the pathogen. Look at the right side, please. I mentioned also specific immune response, lymphocyte, B cells, T cells, and they produce, B cells produce memory cells and plasma cells. T cells, they are different types. They also have memory cells. This is the basic which is asked in the question. Look at that. How vaccination can lead? The previous question, if we go for that, carry out phagocytosis. No, A is a phagocyte because it has a lobed nucleus. It is the mark of identification of phagocyte. If it is a circular nucleus, this one, as in case of B, that is lymphocyte. Take another example. This is C is a red blood cell. D is platelets showing here. So according to the question, phagocyte is only option, and that is A. Clear? Can you move on?
yes, sir.
Excellent. Now, if we go for next question, G, I think so. This Hamna is here with us. Hamna, can you read the next question?
Okay, sir. The diagram shows the circulation of blood in an organism. In which part of the organism is the location of the capillaries in the box?
Just a minute. G, if you look into this diagram, I make it a little bigger, which is just read by the student. Look at that heart, capillary. Oxygen enter, carbon dioxide moves out. Then again, heart, capillaries. Carbon dioxide enter in the capillaries, oxygen moves out. So here he's asking for the box. oxygen is entering, it's mean it is lung, and carbon dioxide is moving out. So it's lung section. For example, if he will not ask this question, he will twist this question and asking this question that what is this box? Then our answer will be, student, human body, because human body is going to release carbon dioxide and it is ultimately the heart is passing on, carbon dioxide is entering into the capillaries, and the oxygen is releasing out. So this will be the answer. Although there are two totally wrong options which are not included as far as the availability of oxygen and carbon dioxide is concerned. Fish body, fish gills. So he's not discussing about that. Secondly, mammalian lungs, capillary wise, they are taking oxygen and producing carbon dioxide. Gee, we are taking the next question. Do we have Hassam here with us? Hassam.
Yes, sir.
Thank you. Hassam, question number 18. Over to you, please.
What is the
Yes,
part
please.
of the definition of transpiration?
Very good. You know, transpiration, as I told you in the previous discussion, that it is the evaporation of water from the mesophyll cell or from the leaf surface. Then, so most closest option, as I told you, it is loss of water from the leaf through the surface of the mesophyll cell. And there is no chance, if you look at the part, the movement of molecules, cell inside, so it is definitely not possible. Movement of particles, cell membrane, region high to low concentration, low to high concentration. So it's a active transport type situation. Transport of minerals from the root into the stem and leaf. So it is diffusion. So there is no option for that three. G, next, yes, who is with us today as well? Ishal is here with us. Would you like to participate? Question 17, Ishal, please.
Yes. Which statement about a function of xylem tissue is correct?
Okay, very good. He's saying xylem. As I told you, xylem in the root stem leaf, its main function is supportive because it is made up of lignin. Lignin helps the plant to anchor in the soil, or it gives mechanical strength to the plant. The chairs, wood which are made of in your homes, they are made up of lignin, they are made up of xylem. Remember, so most closest option, it helps to support the stem and the leaf. So as far as first statement is concerned, they are not accepted because we will reject it because it is carrying glucose, it is for flowing. It is the only transport tissue, no, because there is xylem as well as phloem and are the main tissues, so it is also not accepted. It carries water away from the leaf. As far as the leaf concerned, leaf is taking, phloem is taking materials, assimilates away from the leaf to the other parts of the plant. So it is for the phloem. So he's asking here the question based on that is related to xylem. So our most closest option will be B right now. G, question number 16 we are taking right now quickly, just to take few. We have here with us, other than these, and anybody would like to participate. Fahad Imran is here with us. Would you like to go for question number next, please? Fahad,
Yes, sir.
16.
The diagram shows enzymes and the products of the reactions they catalyze. Which diagram matches the enzymes with the correct products?
Okay, as you know, amylase is specific for sugar. As you know, lipase act on fatty acid fats, so it break it into fatty acid and glycerol. As you know, protease act on amino acid and proteins and forming amino acid. So more closest option, definitely it is not A, it is not B, and it is not C. So which is the most closest is linking, and that one is C. Agree? Can you move on?
Yes, sir.
Okay. Okay. Anybody here also with us right now? Other than these people, those who would like to participate? G. Irab is here. Irab, would you like to participate? Question number 15, please.
The diagram shows a fat globule P in one part of the alimentary canal and the same fat globule Q as it appears in another part of the alimentary canal. In which parts of the alimentary canal are P and Q found?
Yes, fats. You know, fats, basically bile is breaking the fat into small globules, as you know. So as far as the question is concerned, diagram shows fat globules P in one part of the alimentary canal, and the same fat globule Q is appear in another part of the alimentary canal. So it is definitely, it is just a minute. Approximately, some are harder, some are medium, some are easy. Okay, MCQs both the cases are like that. No need to worry. Okay, now as we move on, P, duodenum, stomach, dobara piche to nahi aa sakta. It is not coming back again. Remember, because P first and then Q. So look at that. Duodenum is after stomach. So it is not possible. Same is the case. Ileum to esophagus, because it is again returning back, so it is not acceptable. Because large molecule further break down to small one as the food passes on. So esophagus to ileum, it is possible because esophagus, then stomach, then small intestine, ileum, duodenum, ileum, jejunum. But right now we are checking the last as well. As far as the last part is concerned, because in the rectum there is no digestion, so it is totally again not accepted. So more closed option is C, esophagus to the ileum. Okay, student, these are a few question area which you have to go through. I have already uploaded this in the resource pack. I hope so everybody has the resource, the latest LAC resource pack.
Yes, sir.
Fantastic. Now we are going back again with the next topic after digestive system, and that is circulatory system or transport in human. The next session is going to be, we are here opening session 3 related to that is related to transport in human. So as the question which was in progress in the past paper section, that is phagocyte, lymphocyte. Phagocyte have a lobe nucleus. You can see the diagram above as well. This is the diagram of a phagocyte, and this one is the diagram of a lymphocyte. And again, the WBC, as I told you, they transport oxygen and the carbon dioxide in the beginning. Then phagocyte, they engulf bacteria or destroy the cell wall and ultimately their plasmid. Lymphocyte, they produce antibodies or chemicals which attaches with the bacteria membrane and capture it, and ultimately they destroy it. Or secondly, they also trigger other lymphocyte to make more and more memory cells. So if that pathogen comes again, it should be tackled and destroyed. These are the platelets at the end. Platelets, they are basically related to wound healing, or they produce enzyme thrombokinase, which convert, if you look into the next part of this page, they produce thrombokinase. Thrombokinase convert prothrombin into thrombin, that convert fibrinogen into fibrin threads, and ultimately wound healing takes place. There is the next important part of the same chapter is heart structure function. Remember, some question based on that, they will ask you to label the structure of heart or arteries, veins, capillaries, which is oxygenated, which one is deoxygenated. So all of them I'm just highlighting in short. Look at this one. If we see that there are 4 main chambers of heart, and right at the left, right atria, right ventricle, left atria and the left ventricle. This is our right side of the heart shown, and the left chamber, left atria, right ventricle and the left ventricle. Now questions, sometimes they are asking which vessels are oxygenated, which one is deoxygenated. So try to be very much vigilant. Upper and lower vena cava brings deoxygenated blood. This is pulmonary artery, which is taking the blood to the deoxygenated blood to the lungs for oxygenation. Remember, from both the lungs, oxygenated blood enters through the pulmonary vein, which enters from here into the left atria. Remember, here they pump into the left ventricle, remember this thing, the most powerful chamber of the heart is the, just to color change here right now. It is the left ventricle because it is pumping the blood through aorta to all systems of the body. So that's why you can see this aorta, which is taking the blood up as well as moving it down as well. Bar say it makes an arc and it is going to take the blood to all ultimately system up and downward as well. So it is providing oxygenated blood. Here is very important area. Only A grade student, they are going to handle that. Take a look, please. There are 4 valves inside the heart. In the right atrial, right ventricle here, there is a tricuspid valve. In the left atrial, left ventricle, there is a bicuspid valve, also mentioned here. Tricuspid, which prevent the backward flow. Bicuspid, which prevent the backward flow. Inside the pulmonary artery, there is also a semilunar valve. And inside aorta, there is also a semilunar valve. So these are 4 main valves. Remember the pathway. It's very, very important just to make some color change in blue. It is better. And no, blood from upper lower vena cava, they enter into the right atria, then right ventricle, as I told you, pulmonary artery, lungs for oxygenation. After oxygenation, it returns back to the left atria, left atria, then into the left ventricle. There is a bicuspid valve which prevents the backward flow. Then the left ventricle, which is the most thickest muscular and powerful chamber, it is pumping the blood through aorta to all systems of the body. Remember this thing, questions are based on that, and they will ask you about chambers, valves, sometime related to question transport in human, question related to double circulation. So blood leaves the heart twice, one to the lungs for oxygenation, second to the body system. So pulmonary system or systemic system, these are the 2 circulations which are known as double circulation. Now, blood travels inside the blood vessels, so it's a closed circulatory system. Remember, some questions are based on arteries, veins, and capillaries. Oh my God. Okay. Student, I gave you the MCQ sheet. It has 15 worksheet, 15 MCQs. Try to please finish that all. Takes 5 minutes on that. 5. Then after 5 minutes, we will be back again. Okay. Because it is asked to be, there should be a little break inside. It's a very long time students are studying. So I'm giving you 5, 10 minutes, quickly go through and solve that MCQ sheet, which is worksheet 1. Then we will continue from here, cardiac cycle or arteries, veins, capillaries. Okay. Any question, you can put it in the WhatsApp on your group, and then I will discuss that afterwards. So it's a 5 to 10 minute break. Take your time. No problem. Yes, Hassam, any question? Those students who want to get break, please keep taking the break. If you have any question, you can ask me. Ji, Hamda, Ishal.
No, sir. Everything is there.
Okay, it's fantastic. And okay. Because some students, they want little break. So it's a break for 5 minutes. But during this break, I will show you some short animation. Now take a look of animation. We are not moving further, just an animation is going on. Okay, no problem. We will start after this animation, no problem. There are some, as far as heart is under discussion is going on. Take a look of this short animation.
The heart pumps blood around our body.
Please, everybody is watching the animation of heart structure.
Yes, sir.
Awesome.
Here is a sheep's heart. The heart is made of muscle, so it needs its own blood supply. Here are the arteries and veins. the heart is actually 2 pumps, not 1. The right-hand side pumps the blood from the body and sends it to the lungs where oxygen is added and carbon dioxide is removed. If you squeeze the heart, you can see that the softer muscle is on the right-hand side. The stronger muscle is on the left-hand side. The deoxygenated blood from the body, the head and the limbs comes into the heart here, through this vein, to the right side of the heart. The blood is pumped from the right side of the heart to the lungs through this artery. The oxygen-rich blood from the lungs comes into the left side of the heart through here. Blood comes out here through this wide artery called the aorta. Note how wide it is and that it stays open by itself. The oxygen-rich blood goes to the liver and kidneys and so to the rest of the body.
So this was a short animation to make a little break as well, just to show you overall picture. Now, sometimes the question comes about
Heartbeat is detected in medicine in many ways.
About heartbeat, and there is a very important question related to this heartbeat, or practical question sometimes asked in the paper about ECG. So look at that, how it's simple to calculate the heartbeat at a normal person, with a normal person and the person after doing exercise.
One way is to take a pulse. Do not use your own thumb or finger, as it has a pulse of its own. You could count for 15 seconds and then multiply by 4 to get the beats per minute. First, let us see what the pulse rate is when resting. This pulse was 16 beats in 15 seconds, so that's 64 beats per minute. Now let us see what effect exercise has on the pulse rate. First of all, let's send our presenter off for a bike ride. When he comes back, we'll take his pulse again. Our presenter is back and we'll take his pulse. After the exercise, the pulse rate has gone up considerably. It is now 31 beats per 15 seconds.
Look at that. After exercise, this is the beat, which is 31 beats per 15 seconds. So it's mean per minute you will multiply it by 4 times and automatically it will be 120. I am also sharing it on the group. So you can also measure your pulse rate by doing so your exercise at your home place. Now we are back. This is about the heart brief practical related to. Now we are back with the same topic which was under discussion. Just to confirm from you, ECG, are you watching ECG, heart structure, cardiac cycle? Once again. Please confirm, anybody. Are you watching cardiac cycle, ECG, blood vessels?
Yes, sir. Yes.
Fantastic. Now, cardiac cycle, basically, at ECG, it's a very important and very hard question, basically. Just to take you to this question and showing you how ECG works. What is ECG? How can you handle this ECG question? Everybody is watching ECG assignment now?
Yes, sir.
Fantastic. Look at that. A single person, ECG is shown here in the rough sketch, and it is the atria line and the ventricle line and the aortic line. 3 lines are there in general, and The time of the ECG is normally 0.8 second. One cardiac cycle of a person is 0.8 second. And if you look at that, okay, but I will show you some question. There are questions in the ECG, in the GCE, there is a question in the practical paper, in the MCQ paper. That's why we are discussing quickly within a minute, okay? So ECG is the left side of the heart, which is showing, look at that, atria systole, and ultimately it is a straight line. Ventricle systole, and ultimately it is a straight line. This is aortic line. Remember. Now here on this place where I am putting some circles, very important. On this place, just to change the color. This is bicuspid valve closure. Here bicuspid valve open. Because it's the left side, that's why left side there is a bicuspid valve. Second, in case of semilunar valve, in the aorta, here semilunar valve open, here semilunar valve close. So this is for the ECG main areas. It's been when the atria systole, atria contract, the blood pump into the ventricle, and ventricle line is very high, this one. And look at that, bicuspid closes here, then semilunar opens here of aortic valve, blood pump into the ventricle, ventricle after pumping the blood, the semilunar valve close here again, and the bicuspid valve again open to receive more blood from the lungs. The total time of the cardiac cycle is 0.8 seconds. They will ask you, how can you measure the heartbeat per minute? So 60 seconds in a minute divided by 0.8. So you will divide 60 seconds divided by 0.8 because one cardiac cycle is 0.8. So it will be 75 beats per minute. Remember. Secondly, some questions are asked why heart muscle does not rest. Remember, heart of between 0.8 second, from 0 to 0.8 second, heart muscles, they are going to take rest of 0.3 second. This is the working time of the heart from here to here, 0 to 0.5, and from 0.5 to 0.8 is the resting time. That's why you have not heard from someone, my heart is exhausted. No. We are tired. Our heart is tired. No. Because heart take rest out of 0.8 second. They take rest of 0.3 second, 0.5 to 6, 0.6 to 7, 0.6 to 8. This is the resting time of the heart. Remember, this is for ECG from GCE. Now, as we move on, we are moving back with the next. The arteries, veins, capillary question sometimes comes. Are you watching the slide once again?
Yes, sir.
Thank you. Arteries take the blood away from the heart. Vein take the blood towards the heart. Capillaries, thanks. Capillaries, they are going to diffuse the blood into the tissues. They have large surface area volume ratio. Arteries all contain oxygenated blood except pulmonary artery, umbilical artery. Veins all contain deoxygenated blood except umbilical vein or the pulmonary vein. Remember, arteries have small lumen, thick muscular wall. Vein have large lumen and less muscles. So these are the main areas of arteries, veins, and capillaries. So we are moving further to the next part related to, we have gone through lymphatic, this phagocyte, lymphocyte. Now, as far as the next topic is concerned, disease pathogen. Pathogen is a disease-producing organism. Remember, the transfer from one host to the other host by different agencies, by different methods, or we can say by different vectors like malaria, plasmodium, mosquito is transferring. Look at the top right, body defense, as we are discussing, chemical defense or chemical, mechanical, and our body has three main line of defenses. Remember, one is the skin. Just to make it easy for you, just right now here, our line of defenses are Number one is the, we can say, first line of defense, and that is our skin and mucus. You know, our skin is 99% protecting ourselves from external pathogen, external hazard. Mucus, our respiratory tract, our reproductive tract, our digestive tract, they are with the mucus. Our eyes are releasing continuously sodium carbonate, sodium bicarbonate, and disinfecting the pathogen in the air directly. Our eyelids are continuously moving up and down. In the same way, our excuse me, our skin is releasing sweat, which contain sweat is also disinfecting the pathogens, spores, fungus in this way because they are acidic in nature. Our stomach inside containing HCl. So this is our first line of defense as far as skin and mucus is concerned. Second line of defense is, we can specify here, is non-specific. Now, what is this non-specific? As you have gone through in the previous lesson, phagocytes or inflammation. Inflammation and phagocytosis. So these are, inflammation is the swelling of the area wherever there is pathogenic enter, that place producing chemical, and that chemicals in that area increases blood supply, and WBC approaches there and engulf the pathogen. This is our second line of defense. Third line of defense is highly specific, and in that case, our lymphocyte work. The lymphocyte, they produce antibodies and chemicals. These antibodies are highly specific. Remember, if there is a TB virus, if there is a TB bacteria, if there is HIV virus, if there is a COVID-19 virus, if there is a cholera bacteria, if there is any specific bacteria, virus enter in the body, even a malarial pathogen, plasmodium, they are highly specific to that. This is the meaning of this. If there are millions of pathogen enter, our body has the capability, the Creator Almighty has given this capability to the immune system that we can be able to produce million types of antibodies against that particular pathogen. So they are highly specific. This is about the body defense in short. Now, as we move on, antigen. Sometimes what is antigen? Antigen is any foreign pathogen. Or as we move on, our body has active immunity and passive immunity, and you know, our body defense mechanism directly produces B cells, T cells. So these are our active immunity areas. And passively, if we say, so we have some passive mechanisms, and in that case, you can see from mother to the fetus through placenta, as well as through breastfeed, from colostrum, their antibodies are transferred during breastfeed. So they are passive mechanism of passive immunity. It is. Now, if any pathogen enter into the body, remember, our body develop a memory cell against that. Whenever that pathogen come again, after 1 year, 10 year, 100 year, even any year of the life, it comes. So that memory cells, remember, they identify that pathogen. So memory cell means those cells which already target the particular pathogen. So they stays inside the body. So as we move on, next topic is gas exchange. Remember, ask you will ask you to label that respiratory system relevant to nostril, larynx, pharynx, trachea, bronchus, bronchiole. Now here the question based sometimes intercostal muscles. In between the ribs, there are two types of intercostal muscles: external intercostal muscles, internal intercostal muscles. When we inhale, remember, externals are outward. For example, this is the ribs. These are the ribs shown. For instance, I'm just showing you two ribs. Remember, there are 10 ribs attached on the front with the sternum, chest bone, and there are 12 ribs on the back, 2 extra, which is protecting the kidney. So there are overall 12 ribs, but 2 on the back, they are not coming forward. They are only protecting the kidney. They are incomplete. But 10 ribs attached back and forth, vertebral column to the sternum on the front. So back and forth, they are fixing. Now, as far as the muscles are concerned, take a look. The muscles which are outward, here I'm just giving different colors or in easy way. For example, these are the external intercostal muscles, outward, and the muscles which are inward, for example, inside, they are known as internal intercostal muscle. So when we inhale, the external intercostal muscle, as mentioned, these external intercostal muscle, they contract, and internal intercostal muscle relaxes. Chest move upward, outward. Volume increases and pressure decreases. Diaphragm become flat. So this is during inhalation. But when we exhale, the reversal is done. You will specify the reversal in your answer if it is going to be asked, because mostly the questions are based on that, or alveolar structure. Now take a look about the alveolar structure. Sometimes they are asking you what is happening what is the amount of oxygen in the breathing in air, breathing out air. So it is 21 in oxygen in and 16 when it is going to go out, oxygen. Carbon dioxide 0.03, 0.04 during inhalation. Carbon dioxide 4 to 5% during exhalation. So these parameters, diaphragm contract in case of inhalation, as shown in the diagram. They are comparatively above and below. So inhalation way, it is going to be compressed downward. It is going to be, if you look at that, it is going to be little down. So if it is exhalation, it is become, you can see exhalation takes place, it is forming a relaxed condition or a dome shaped structure. There are some areas which are asked in the chapter of respiration about aerobic and anaerobic respiration. If you look at this structure, look at this equation. Glucose into lactic acid. In the muscles, when anaerobic respiration takes place, glucose is converted into lactic acid. In case of plants, glucose is converted into ethyl alcohol and carbon dioxide. This lactic acid inside the muscle, for example, you are daily doing some exercise, 30 minutes, but one day you do exercise 1 hour or 2 hours, more than that, it will develop lactic acid. It will cause muscle fatigue, muscle cramp. So to convert that lactic acid into carbon dioxide and water, which we call oxygen debt, this lactic acid, which is accumulating in the muscle, it will cause muscle fatigue, muscle cramp. Remember, to convert that lactic acid into carbon dioxide and water, the amount of oxygen required, which we call oxygen debt. Now, what is oxygen debt? It is the amount of oxygen required to convert lactic acid into carbon dioxide and water. Although with a little amount of energy. So this is for a question which is going to be asked in the paper about anaerobic respiration, the muscles, what happens. So you will specify in such a way. Next is in case of plants, it is forming ethyl alcohol and carbon dioxide. No doubt, aerobic respiration generates huge amount of energy and aerobic respiration. But in case of anaerobic respiration, just to make a comparison of that as well, take a look. For example, if there is 36 molecules of ATP is produced in aerobic respiration, so in anaerobic respiration, approximately we can say 2 are produced in anaerobic respiration. So this is the ratio, or we can say 18, in short, we can say 17, 80, 60, 70, around about 18:1 overall. Clear? Aerobic generates huge amount of energy as compared to anaerobic respiration. Mostly just to take you some of the question, because we have recently gone through one of the topic, that is circulatory system. Take a look, a little change in the how the question come in the paper. Look at that ATP paper question. Everybody is watching the question of ATP?
Yes, sir.
Fantastic. Now he is showing you an artery. It's lumen, thick muscular wall. The question about magnification. Remember, you will use simple pencil, HB pencil, and no need to go for shading, no need to go for all that creativity with respect to sketching. It's simple rough sketch you will make as you are watching. Your diagram should be little 1 to 2 times bigger than the diagram which is shown. For example, here you will make the diagram, and he's asking here only draw the diagram, showing the layers. The question how you will handle that, thank you, how you will handle that question, take a look. If the above question is asking the layers, for example, you will make the layers of the arteries, at least you have to mention 3 main layers. So there are 3 main layers of the, because he's asking show the layers of the artery wall. So if you just look at back the layer shown, internal layer, which is little rough because blood pressure is there, that's why it is going to be little, there are some little wrinkling inside the layer, it is not smooth. So this is, you can see this is with a wrinkling shown here, a little rough surface shown. This is your one of the layers shown. Remember, I also put this thing in my this resource pack at the end also. Second layer, the middle layer, which you will study in the higher class, tunica media, then the third layer. So you will make 3 layers of artery as shown here, as far as the question is concerned. Secondly, question. Use the ruler, put on A and B point, find the measurement, write it here. Length of AB is, for example, it is 20 millimeter. Remember, the length is 20 millimeter, for instance. Remember, also mentions as units in your answer. Formula is already given. Magnification is equal to image over actual size. So if you simply put the, he's asking here, calculate the actual size. So when he's asking about to calculate the actual size, at that time, you will simply take actual size is equal to magnification. Here you will write image size over magnification in short. I guess I'm writing in short, or over you can see the magnification. And then you will put the values. He has given magnification here. Look at the diagram corner 13. So your image size 20 millimeter, magnification 13 over G. How much G Fahad? How much 13 over just it is 20 over 13. Yes. G, how much? Yes, anybody? Calculator? Ria?
Sir, 1.53.
Very good, Hassam. Perfectly right. So you will put here the value of actual size, and as mentioned, they will, if they have given you some space here, no. You will write here, show your working. You will show this working all. Remember, don't write directly the answer because he's asking here, show your working. It's a 3-4 marks question. Very good. Fantastic. Round figure is 1.54. Perfectly right. Now, as we move on to the next, a very simple question. What describe one similarities, one difference between the artery and one in the vein. So one similarity and one difference. Artery, vein, if you look into the diagram, both have lumen, right? But difference, both lumen sizes different. Small lumen, large lumen. Yeah, regular lumen, irregular lumen, large lumen. Or you can write arteries, thick walls, veins comparatively less thick walls. So this is your difference. He's asking for, remember, he's asking for the visible difference. Sometimes student is so hurry and they write oxygenated, deoxygenated, valves present, valves absent. No, please try to be very much vigilant because these questions are related to visible differences. Now we are back again with the. I have already placed this thing in the resource pack. How can you draw the diagram? Look into the, if you go on the page, I hope so everybody has the resource pack, LSC resource pack.
Yes, sir.
Very good.
Yes, sir.
So, fantastic. If you go on page 43, you will find the same question which was in the past paper. I make the diagram for you. You can make 3 layers of artery just like that. Just like that. 3 layers. If they will give you a question based on the leaf structure, how you can handle that? He's asking make layers of leaf structure. You will not show inside the tissues. You will not show inside the cells. You will show only the tissue bare layers and not the cells in that. This is the meaning of the question. Now take a look of another question which is asking here right now. I put it here on page, yeah, page 59. Another question taken from some other year, 2020 somewhere. A photomicrograph shows a cross section of the leaf. Yes, any question? Thank you. A to B, magnification is 100. Draw a diagram, show the layers present in leaf structure. Now he's asking to make the diagram here. You are going to make 5 layers and the vascular bundle. As you have seen in the previous diagram. Remember, it's a 4 marks question. Then you will measure the length from A to B. For example, it is 40 millimeters. Magnification is here. What is asking the next question? Take a look. A to B length, as we mentioned, it is 40 millimeter. Magnification he has already given. So we will simply change the actual size out. Actual size and is equal to, and we simply put here image size over magnification. Now we are putting the values here. Our image size that was 40, agree. And how much was the magnification with the diagram? Anybody?
100, sir.
100. Thank you. Yes. Yes, please. What will be the actual size?
0.4.
0.4. Perfectly right. We are here. That's right. That's correct. Thank you. No, wonderful. We are moving on here to some other next part of the next chapters relevant to that. Okay, I get another message from the office. Okay, I'm sending you here just to giving you 10 minutes more for the next worksheet because according to the session 3, we have 5-10 minutes for the short quiz. Although I'm putting another short worksheet on the group right now. Try to please see it again. Please take 10 minutes to work on this worksheet again. Yes, over to you. Quiz test 2. Yes, you have take 5-10 minutes. We will discuss afterwards. We have here a student give me a message about GCE ATP paper. Let's take a look now. As far as those who are taking GCE, please have an eye upon the slide once again. Uh, it's a GCE ATP paper. Take a look. He's showing you those who are taking GCE. Even the other people can also see that because the syllabus is same as far as this question is concerned. He's giving you a question, straight investigation of the growth of some of the seedling. They had 3 dishes of seedling ABC have been grown in the same condition for different amounts in time. The selected 3 seedlings from each dish measured at their height ultimately to the nearest millimeter. The resulting dish from A to C is shown here. A is done for you. Mean you will write. Add that 3, you will write the mean. Mean is 23. I think so. First one is mean, very clear. Second is not given anything. Third, 2, 4, 3. Its mean is 3. I hope so. Up till here is clear. First one mean is 23. Agree? Third mean is 3. Agree? Now, what is the value of B? Take a look. He has given you a diagram. Seedling A, seedling B, seedling C. You will measure with the help of a ruler and putting the value here. For example, just to give some examples, it's 10 millimeter, for example. Next one is, for example, it is 14 millimeter, for example. Or third one is, for example, it is in less than that too, it is 8 millimeter, or we can say 6 millimeter. Roundabout, it's 6 millimeter. So you will add that 3, you will write the mean. So mean, how to calculate the mean? Add the 3, divide by 3. Okay. Next. Use the data suggests the seedling had been grown for the longest period of time and give the reason of your answer. The seedling grown for the longer period of time. Definitely more nutrition, more oxygen, more minerals. So it will grow for the longer period of time. More environmental conditions are favorable, or high vigor plant, high vigor seed. These are the parameters you can specify. I also said this on the group as well. No, he's asking about little bit about the graph, please. Look at the graph. This is related to basically P6 germination chapter. Growth for 6 days, mean height is 8. Growth for 10 days, mean height 23. Growth for 12 days, mean height 27. Look at that. These parameters on the left side, these are the independent variables. And the other one is the dependent variables. So independent variables are always taken on the x-axis because they are the causes which can be manipulated. These are the causes. And dependent variable are the effects which are taken on the y-axis. Remember. Now here you will put the values of 6, 10, 12, 15, 20, 23 here on the x-axis. Right, student? Here you will put the values of 6 in the same way, 10, and the same way properly displace that all. The same way 12, and in the same way 14, and ultimately 16, and go for up to 20 as well. So as far as the y-axis is concerned, we'll put the values of y-axis. It is how much they are given. Oh, he has given, just a minute. Y-axis values are 8, 23, 27, 32, 36, 37, 8 to 40 approximately. So here we will put the values 8 to 40. It is good to take that, for example, it is over 0, for example. starting from 0, and this one is, for example, 10, the same way 20, and because it has gone up to 38, that's why we can move up to. Please do the thing very straight way with the pencil, x-axis, y-axis label, and then you are going to make that. If it is a best fit, please attach that all or circle that all, make the cross that all. For example, you get the best fit, for example, right? So wherever your values are, put the circle or the cross on that. If they are on that area, circle or cross on that, like this. Or you can circle them as well. Both are right option. Use the pencil for that and make your work very neat. Remember, it's a bonus box. Bonus box in the graph, bonus box in the diagram, formula, and so on. Okay, he's asking some question based on, okay, a little bit about, okay, one more thing. This design and investigation to determine the effect of pH of water on the growth, mean height of the seedling, which you use seedling grows in a petri dish in the laboratory. 6 box question. Operators, independent, dependent variable, method, procedure, safety precaution, conclusion. This is your parameter. Remember, your main, we can say, the flow of the question should be, the flow of the investigation should be like that. Right? Here you will take 5 petri dishes, for example, or different pH, for example, 2, 4, 7, 20, 2, 4, 7, 11 or 14. 5 petri dishes, in put a cotton wool, same amount of water, same mass of cotton wool, put 20 seeds in all the, soak the seed before you put it in. So these are the parameters which you will specify. Then here, that is, or remember, in germination, no need for light, water, oxygen, and air. Water, oxygen is the most important thing. 3 main necessary conditions. One is water, second is oxygen, and third is temperature. So in this, you will specify 3 main conditions. Your temperature of the room, it should be same because he's asking for the pH. So you are working on pH. You will measure their growth within 1, 2, 3, 4, 5 days and specify in your answer at which pH the growth of the seed are going to be better. Right? This is GCE question, remember, paper, because there was a question asked someone. So just to tackle that as well. Now we are back here with the just to confirm, everybody is watching the slide, please.
Yes, sir.
Fantastic. We are back here with the last time we were here with the disease respiration. Now next, excretion. Ji, are you watching the slide of excretion? Ji, Hamda is here. Are you watching the slide of excretion?
Yes, sir.
Excellent. Thank you. Now, excretion is the removal of nitrogenous metabolic waste material. Definition can be asked to you. Then labeling of the diagram, past paper question based on that. Just to take you people to the one of the question, because here, just a minute. A brief introduction, then I will take you at the end of this session 4 for the question. Look at that outer cortex, internal medulla with the pyramids, 16 to 20 in number. Internal depression is known as renal pelvis. All the way ureter is coming out and going down into the urinary bladder. Look at that on the left side as well. Both kidney release ureter into the urinary bladder. From the urinary bladder, it is going out through urethra. So this is overall description. Now question directly come on the unit structure of a nephron. Kidney unit structure. Thank you. The unit structure is a nephron. Just like Alveoli is a unit structure of respiratory system. Red blood cell is the unit structure of circulatory system. Villus cell is the unit structure of a digestive system. Just like that, nephron is the unit structure of a excretory system. No, this is details are not in GC, but IGCSE details are there. Okay. But here in case of IGCSE, this whole labeling you should know. Number one, nephron has three main parts. Remember, one is the renal capsule. Number one, look at that. It is a renal capsule, which has bunch of capillaries inside known as glomerulus. Bowman's capsule with glomerulus, bunch of capillaries. One artery enters, one artery leaves. Artery entering, artery leaving. Entering known as afferent artery, exit artery is known as efferent artery. Just to write in short, afferent artery or efferent artery. Afferent artery. Then efferent artery. So from afferent artery, blood enters into the glomerulus, where it is filtered. So we call this filtration ultrafiltration. Look at the bottom, ultrafiltration. What is ultrafiltration? Basically, it is high-level filtration in which everything from the blood enters into the Bowman's capsule except large protein molecules and blood cell. They will not enter, they will come out. So large protein molecules, they will not enter and they will come out. Now, in the second part, which is known as renal tubule, it has three parts: PCT, loop of Henle, and DCT. In the second part, which is known as PCT, loop of Henle, and DCT, selective reabsorption takes place. Selective reabsorption means to say useful substances like glucose, amino acid, they are reabsorbed back. If I take you to the question which is going to be asked in the past paper based on the excretion section. Now, this is a past paper question which is putting you label the structure. I'm sure everybody is watching the slide, please.
Yes, sir.
Okay. K, cortex. L, medulla. M, renal pelvis. P, renal artery. Q, renal vein. N, ureter. Remember, they will ask you to label that. Or sometimes they are putting a question, again, the repetition of the same question in another way in the new year. Or as far as the, okay, nephron. Nephron question, look at that. This is a past paper question from the nephron section. Define the term excretion. It is the removal of nitrogenous metabolic waste material. For example, kidney, and example is urine. You can specify. Now, what is, if you see, one, it is afferent arteriole, which is taking the blood into the glomerulus. Two is the Bowman's capsule. Remember, just two. One is afferent arteriole. Two is Bowman's capsule. And three is PCT, proximal coil tube. And four is collecting duct. So just to make it little clear, afferent arteriole, and two is Bowman's capsule, or we can say renal capsule. And three is PCT in short. It is also accepted. This abbreviation is also accepted. PCT, proximal coil tube. And fourth is the collecting duct. So as I told you, in the first part of the nephron, there is ultrafiltration here in this place. And in the second part, here, selective reabsorption. Useful substances, they are reabsorbed back into the blood vessel and goes back through renal vein. Last part of the nephron is known as collecting duct. This collecting duct, basically, it has a very important role. Take a look. From the pituitary gland, a hormone is released and attaches to the collecting duct. Now, what is the role basically? Collecting duct function is osmoregulation, water balance. Our pituitary gland produces a hormone which is known as ADH. Now, when our body is in need of water, remember, ADH attaches to the collecting duct membrane and absorb more water. More ADH is produced by the, I'm writing it short, pituitary gland, PG. More ADH is produced by the pituitary gland, attaches to the collecting duct, more water reabsorbed. I'm showing with the arrow, water is coming out, it is reabsorbed. On the other hand, urine which is forming, it will be more concentrated. And it is at the time when the person is doing some, we can say, work, too much work, or we can say when there is a lot of sweating, person is losing water, then the pituitary gland produces ADH. More water is reabsorbed and urine becomes concentrated. On the other hand, if person is not sweating, not in the exercise mode, so less ADH is produced, less water reabsorbed, and urine becomes dilute. So this is the function of collecting duct. If you look at the remaining question, which is coming ahead, just to take a look of that. He has given different concentration, increased concentration of different substances. They will ask you role of the kidney in maintaining concentration of blood plasma. So kidney has two main functions: one, blood filtration; second, osmoregulation. This is one of the worksheet from the past paper. Another question from the past paper of the nephron. Take a look. Gee, I would like someone to participate. Gee, Dr. Fahad. What is A, B? What is X, Y, Z? Yes, anybody? Hassan? Anybody? Ishal? Hirab? Or Hamda? What is A? Okay.
Glomerulus.
Correct. It's glomerulus. This question is from IGCSE. Yes. So what is B? Anybody can participate. We have just recently done. It's PCT, proximal coil tube. Okay. Label Z. Collecting duct. Okay. Everybody. X. X is okay. Yes,
The
yes.
artery.
X is afferent arteriole. Okay. It is artery, no doubt. It is afferent arteriole. Remember. And A is a glomerulus. So I will send some assignment on the group as well if you want with the MS. But right now, because of the scarcity of time, we are moving to the next part, coordination response. Yes, everybody is watching the slide. Please confirm.
Yes, sir.
Okay. In this chapter of coordination response, you have three topics. One, nervous system, sensory motor relay neuron, brain. Secondly, you have inside that eye structure function. It's near and distant vision area. Third is the hormone section. So we quickly go through these areas up till today's time, which we have. Excuse me. As far as the neurons are concerned, remember there are three main types of neuron, and electrical impulses travels in the form, the messages are traveling in the form of electrical impulses or charges. This is the cell body of the neuron. On the left side, please take a look. A cell body. And this one is the axon fiber. Message always travel, electrical impulse always travel from cell body to axon. Remember, it will always travel from cell body to axon. So when we say motor neuron, so motor neuron take impulses from the sense organ to the spinal cord. Motor neuron, in brief, they take impulses from sense organ to spinal cord. Remember, then sensory neuron, this is the first definition is for sensory neuron. Remember, I'm just making it down as well. Oh. No, this is for the sensory neuron. They take impulses from sense organ to the spinal cord. Motor neuron, they take impulses from, or responses we can say, from spinal cord to the sense organ, to the effectors. So this is the main difference of sensory neuron, motor neuron. Sensory neuron takes impulses from sense organ, any five senses, touch, smell, taste, hearing, up to the spinal cord. But motor neuron, they take impulses from the spinal cord up to the effectors, and effectors which we have, that is muscles and glands. Remember. No, relay neuron, they are only present inside the brain and spinal cord. And their main function is interpretation or translation of the message. Remember this thing. Now, this is about a brief function. Now, as far as their generalized structure, look at the structure. Motor neuron has a cell body, long axon. Sensory neuron has a long dendron with cell body and a short axon. Remember, because on the right side, you have a good view of sensory motor relay neuron. This one is a sensory relay neuron. Look at that. Starting from receptor, it is a long dendron with the cell body, making a bunch of neuron here, dorsal root ganglia, and axon fiber. This one is relay neuron, which is inside the spinal cord, dark matter also. And there is again cell body of motor neuron up to the effector. So they will ask you to label that dark matter, white matter. Dark matter has more cell bodies of relay neuron. In short, we can say, it is better to write relay neurons as well as motor neurons. They are inside the dark matter. They are cell bodies, remember. And sensory neuron cell bodies outside the dark matter. Another important area, it is dark because it has nucleus, cell bodies of relay neuron, and the motor neuron, they have nucleus, nucleoplasm. That's why it is darker. But there is myelin sheath, these areas where I'm putting circles. So that's why it is whiter, because it's a fatty sheath. So it is whitish in color. Synapse is the gap between the two neurons. Remember, it is the gap between the two neurons is known as synapse. Look at this gap here. Sensory and motor. Look at the gap here. Sensory and relay, the gap, first one, and then relay and motor, the second gap, which is known as synapse. So electrical impulse convert into chemicals, which are known as neurotransmitters. So message is dispersed from sensory to electrical impulse to chemical impulse in the form of neurotransmitter, and from again neurotransmitter into electrical impulse. If we make this in a way like this, it is better. This is electrical impulse on the sensory neuron. In the synapse, it is chemical again. Again, relay neuron, electrical impulse. In the synapse, again, it is chemical. And again, in the motor neuron, it is electrical. And ultimately, the effectors are muscles and glands. They show the response. If brain is not involved, we call it as reflex action. Remember, if brain is involved, so we call it as a voluntary action. Now, on the basis of that, questions are coming mostly. Now, we are moving to the next part of this chapter, that is eye structure function. Eye has three main layers. One, internal layer is known as retina. Second layer is known as choroid. Outer layer is known as sclera. Remember, outer one, sclera. Second one, choroid. Internal one, retina. So to make it easy, it's very simple. Outer one, but it is good to go like this. RCS. So it is the easiest way to memorize. RCS. Choroid, internal layer, which has, retina is the internal layer, which has two types of cells, rod cell and cold cells. Look at the function one by one quickly. Rod cells, cold cells inside the retina. Here, there is maximum number of cold cells at fovea centrala. Image is forming on it. Image is always inverted. It is real. It is small in size. Choroid, which is supplied with blood capillaries and nerve cells, sensory and motor. Sclera is the outer covering. From the front, it is known as cornea. Pupil is a hole in the iris. Iris has a hole which is known as pupil. Remember, iris is the colored part of the eye. Pupil is the hole which is controlling the light, amount of light. Lens is attached here, which is handled by suspensory ligament, both sides. Remember, and behind the suspensory ligament, there is ciliary muscles. So suspensory ligament directly holding the lens first. Behind the suspensory ligament, there is ciliary muscles. They work antagonistic, just like that. Pupil has also two types of muscles. They also work antagonistic. This thing which we are discussing onward. One thing more, on the image, on the yellow spot, image is forming on fovea. Fovea is yellow spot. Remember, other name is this in the books also. Yellow spot. And here is the blind spot. Here, where no image is found because there is no rod cell and no cone cells. That's why it is known as the blind spot region. So rod cell, they work in the dark dim light. Cone cell work in the bright or highlight or colored light. The main brief areas. Now, as far as the eye accommodation is concerned, distant vision, near vision. Mostly asked questions are asked on that. Let's become thin for distant vision. Suspensory ligament contract, ciliary muscles relaxes. For near vision, let's become thick. Suspensory ligaments relax, ciliary muscle contract. So this is for accommodation. Now, next part of this topic that is related to hormones. Main hormones of your syllabus are first one mentioned adrenaline. Adrenaline is produced by adrenal gland, and its main function with respect to at the time of fight flight it is produced and increase heartbeat. Pancreas produces insulin and glucagon, which control glucose metabolism. Testis produces testosterone, estrogen, and testis produces testosterone, and ovaries produces estrogen, progesterone. Remember, testis controlling the sperm production and male sex characters. Ovaries controlling by the release of estrogen, progesterone. They control female sex character as well as egg production or uterus thickness. On the basis of that, take a look of the question which they have given in the You have the question, which is the latest, which is with you right now. Question number 2. Yeah, it's here. Everybody is watching the question, please.
Yes, sir.
Okay, thank you. The first question of the paper 4 here right now. Look at this question which they gave. Just to take you with the papers as well. That's why I'm also touching the papers as well. Okay, ecology, bacteria. Okay. Yeah. This is the question 2021. G Janab. A. Lungs. B. Heart. C. Adrenal gland. E. Pancreas. D. Testes. The question which we have just gone through in the coordination, and that is asking here, name the endocrine gland C. If you keep in mind the question above with the diagram as well. Yes, C. Name of the endocrine gland. Yes, please. Anybody?
Adrenal gland, sir.
Very good. Adrenal gland. So it's adrenal gland and hormone produced.
Adrenaline.
Correct. That's right. Adrenaline. Perfectly right. It is produced at the time of fight flight. G. Next, insulin is mentioned. He's asking name the endocrine gland. He's here asking for name.
Pancreas, sir.
Correct. Pancreas and or letter.
E, sir. E.
E.
Correct. That's right. It's E. It's wonderful. Yes. Next, testes. Letter.
D.
Perfectly right. Next, hormonal secretion produced by them. What is the hormonal secretion?
Testosterone.
Correct. That's right. Testosterone. Okay. So this is related to endocrine gland chapter up till here. Now, just to move back with the same topic which was in progress. Excuse me. So we are here with coordination response with respect to hormones. Everybody is watching the slide of the hormone, please confirm.
Yes, sir.
Fantastic. Okay. Next is homeostasis. Homeostasis, basically, it is to maintain the internal condition of the body at normal state, at balanced state, for glucose, for temperature. So as far as the syllabus is concerned of both GCE and IGCSE, please remember that this skin structure, they are going to ask you how it maintains the temperature by vasoconstriction and vasodilation. Vasodilation, blood vessels become dilated, blood vessels become constricted, narrow or broader. Now, when the body is, we can say, in need of, we can say during summer or when the temperature is high, blood supply to the skin, it is going to be faster. Heat is radiated out of the body. Hair muscle relax, hair become flat on the skin, and high sweat production during summer. But on the other hand, when there is vasoconstriction, you can see these labeling shown here that blood supply to the skin, it decreases. Heat is preserved inside the body during winter or when there is much colder condition, and hair muscle become, we can say, they contracted, hair become erect, goosebumps during winter, and less sweat production. They will ask you how the body is maintaining temperature. So you will specify vasoconstriction, vasodilation, 3-4 points for that. For each of the case, you will specify in your answer. And it is controlled by hypothalamus to maintain the feedback mechanism. So our body temperature is maintained. Our body, so one more thing, very important. Which two structures in the skin are necessary for, we can say, temperature balance? One is blood capillaries, and the other one is sweat glands. So more sweat is produced during winter, during summer, and more heat is radiated out during summer. So these two structures which are related to temperature control. One is capillaries, blood capillaries. Second is sweat glands. Okay, student. Next, the next chapter is coming ahead. That is drugs, and our time is 5:30. Inshallah, right from here we will start tomorrow, and I will tackle some questions from the past paper for paper 1, MCQs or paper structured, and ATPs as well. If you have any query, you can put it on the chat box or on the group as well. Up till here, any question if you have for today's session, I shall be very glad. Whatever your queries about, or I'm very glad particularly that everybody participated very well and gave answer with enthusiasm. Any question would you like to ask, or what do you feel? I mean to say, they are asking about. I would like to just compile here by this that you have to feel free nowadays, because there is a exam pressure everywhere. Everyone has that. Not only the students, every teacher, even the parents, they are under pressure. So it is an overall effect everywhere. Feel free. Second, try to revise whatever you know. If you will grab that thing, you can be able to do the paper much better. So we will start from here onward tomorrow. Everybody, thank you very much for all your responses in today's session. Sonera and Fahad and Hamna and Hassan and everybody. Thank you very much. Have a nice day. Allah Hafiz. Allah Hafiz, everybody. Allah Hafiz.
Yes,
Have a
sir.
nice day. Allah Hafiz. Thank you. Allah Hafiz.